the next speaker is me. So instead of dwelling on my past, I'm going to talk a little bit about what's going on right now. I am currently, in this moment, the leader for the Stand Up for Science rally that's going on in Lansing, Michigan, that's been going on since noon and will end probably by the time I finish this talk. And I just wanted to say that I find it really important that that's going on. But some of you might be asking, Howard, you skipped your own rally. It's like skipping your own wedding. What are you doing? I thought it was much more important that I be here because I do perceive the people doing racial disparities and health disparities research are under threat right now. The research itself, there's a debate, but I think it's also under threat. And I just wanted to say that I work in Detroit. Detroit is a 78% African-American population city. The patients that we see in pancreatic cancer, 25% of them are African-American. We would not be doing our duty as researchers and clinicians if we were not studying pancreatic cancer in African-Americans. They're the patients right in front of us. So I'm doing this for the science. I'm not doing this as an act of defiance. But if it's going to be interpreted as an act of defiance, I am proud to be here with you in defiance. And right now, let's go to the science. This has got to be the dorkiest title of the day, so I'm just not going to read it. This is going to be work I'm presenting that's largely done, I would say in collaboration, but I'd say led by one of the young PIs that I mentor, Dr. Nina Steele at the Henry Ford Pancreatic Cancer Center, which has recently partnered with Michigan State University. And because of that partnership, some of the work I'm going to present today has been worked out by graduate students in the Kui Lab at Michigan State, Yusong Wu and Aoqi Xi, as well as some other students, one from my laboratory, Daniel Salas Escabillas, and a very talented bioinformatician, also at Michigan State, a graduate student, Ian Lovelace. Everybody else works for me, so they can just take their lumps. So as Amber's already presented a lot of this data, a couple of things that weren't mentioned is that in African Americans, pancreatic cancer is 20% more likely to form. So she focused mostly on survival, which is also worse in this patient population, and something we're obviously very concerned about. Something that I work on in my laboratory is the initiation of pancreatic cancer. So something that I'm very interested in is trying to parse out why this is happening. And I just want to point out also that when we look at the histology of the human pancreas, it's pretty bland when it's normal. A bunch of cells making digestive enzymes, a bunch of cells making insulin and other hormones, and then other cells that are important for transporting these digestive enzymes into the small intestine so that we can eat our food. And then when pancreatic cancer forms, it becomes much more heterogeneous, much more complex. It's made up of a bunch of different cell types, including the tumor cells, which themselves are heterogeneous, but also a huge stromal response that is made up of scar tissue, essentially, so fibroblasts and inflammatory cells. And something about looking at these complex tumors in their natural environment is something we're very interested in doing. So I'm not going to go through that. And as Amber also touched on, we don't need to dwell on it too much, but I'm going to overemphasize it a little bit. She talked about the data and the sequencing databases that is very much underrepresented for Black patients. I also want to point out that the cell lines that we want to use in vitro studies are almost all derived from white patients. Very few. We have exceptions. I'm pretty sure I can speak for Amber and say there's probably some cell lines from those organoids she's been making that are African American, as well as some of the ones that we are making as well. So we're trying to fix that gap. But moving on to this, going on to this omics idea. So single cell sequencing is a hot topic these days, where we're actually going to look at all the genes that are being made by every single cell within a tissue. And so what Nina has done, along with Ian, is compiled all the existing data that has been published in single cell studies, which are generally low and underpowered because they're so expensive to do. So she thought it was important, in order to get statistical significance here, is to compile all the published studies and put them together and look at them and see what she can parse out. Now, what I'm going to emphasize in this data is that when she looked at over 200 patients, donors and pancreatic cancer patients, and did ancestry using a modality that was developed by Al Levin, the talented statistician at Henry Ford. They could define the ancestry of the patients based on their transcriptome, their single cell transcriptome, and looking at the African American patients. Out of 200, there were 3. So this is worldwide data, only 3. We've expanded. She's expanded this atlas to 300 now. We haven't done the ancestry yet, but I'm going to guess we might get to 5. It's just the way it's going. And Amber did a great job of explaining this. The different subtypes of pancreatic cancer, the classical, that with patients with largely classical molecular tumors live longer compared to those with the basal-like tumors. So again, just to go back over that. And the idea here, she's brought up the idea that there are classical, they're basal-like, there's other heterogeneity here, that there's something that some people would call a hybrid, that they're making the genes of the classical and the basal. But again, it's really not that simple. So, and I'll explain what I mean in a moment. So what we wanted to do is spatial transcriptomics. That is to look at the whole transcriptome within the patient's tumor section, and be able to identify if we can find differences in the subtypes in those cohorts. And by the way, Amber, we should have talked before this about this. But because these are so expensive, I'm just going to say right out front, it's a very, very small sample size. We have 6 African-American patients of our own, 6 white. All of these are treatment naive because we wanted to keep that control. They haven't seen chemotherapy. We also pulled out data from the H10 project that had another African-American patient within it and 9 more white patients. So this is our sample size. Again, very small, hard to make really firm conclusions about this, but I want to just give you something to spice it up and show you what we're doing after this. So this is what it looks like. If it looks really pink, that means it came out of the Henry Ford system. If it looks really blue, it came out of a H10. This is just the histology with 2 different sets of hands doing it. So it looks very, very different, but it's all the same. This is just what all the tumor samples look like, particularly the ones that we went in and sequenced. And for those that are not familiar, and this is, I would say I don't know, probably not the first gen, but close to the first gen version of spatial transcriptomics, where a section gets put over a square that has a bunch of different spots that have primers on them, and essentially gives you a whole transcriptome. But the spots themselves are not terribly small. In fact, generally the size would include, I'd say 7 as being generous, but probably 7 to 15 different cells within each spot that's being sequenced. So you don't really know if you're looking at a tumor cell, an immune cell, a fibroblast, or whatever. And then so it makes it very complicated. So what can we do? So going back to the atlas that Nina put together, we can use this single cell atlas, which is just single cells, one cell for every spot we see in these UMAPs, and then use that to deconvolute the big spots that we find in the spatial transcriptomics to identify what cell types might be there and what genes that they would be making. And so what you can see here is overlay after deconvolution, that all the different cell types that have been identified just by color coding. And if you look really close at how these color codes look, they're actually pie charts. So if they're primarily tumor cells, the pie chart will be mostly red. But you can see all of these big red pie charts have a little bit of blue, a little bit of purple. So there might be some fibroblasts there or whatever. And then, of course, some of them are 50-50. But this is the best we can do at this level for deconvolution, and it's actually pretty powerful, we think. So deconvoluting all of these patients that I just mentioned, what we wanted to do was see what's going on with the basal and classical subtypes. So we used a gene list that was identified in the Cell paper a few years ago. So the gene list that identify the classical, identify the basal, and superimpose this on the spatial transcriptomics. And you can see that we can see largely areas that are basal, but other areas within the same tumor that are somewhere in between, other areas that are more classical. So again, very heterogeneous, but going through each one of these patients, we found that essentially every patient has a mix of everything. Some are primarily classical, some are primarily basal, but they are not none of them are all one or the other. So what do we find here? Looking at the classical score, that we thought it was really important that we saw this data. So the non-African American, so mostly white patients, had a higher classical score than the Black African American patients. That is, they had the type of tumor, primarily the type of tumor that gave better survival. So maybe this is part of the explanation for why African American patients don't survive as long as white patients. And this isn't just driven by one patient or another. Looking at each one of the patients individually, you can see the majority of the non-African American patients have a higher classical score than the African American patients. It's very important when you do this really small cohort of samples to have an independent validation, and then therefore a very simple assay that you can actually look for the markers of the classical and the basal. So we're looking at the marker for the classical. This is TFF1. And then go and look at a larger cohort of patients and define whether this data holds up from this relatively small cohort. And you can see that the TFF1 is relatively low compared to what the white patients always have a large amount of classical characters. So this assay suggests that the spatial transcriptomics is holding up. And this is some quantitation for the subset that we've looked at so far, and we're of course expanding this to a larger group. So it was very logical that we went in and said, hey, if the white patients have more classical, then of course the Black patients must have more basal-like. And the answer is that's not true. They have essentially the same amount of basal-like character within this cohort. These are the individual patients. You can see it's a little bit more variability, but it didn't hold up statistically. So this is where I actually got to say something smart to Nina and said, well, if it's not going from classical to basal, it's got to be going somewhere. And so what Auchi did was go through and look at not just the classical and the basal, but defined a gene expression profile for the intermediate co-expressor. This is actually from the previous paper. But what she did was when she went into the cells that were making classical and basal, she found about 50 to 60 genes transcripts that were marking those populations specifically. So I really want to go and say that I don't really think that the intermediate or the hybrid, if we want to call it that, is really that. I think it's its own subpopulation that has characteristics of both, but it also has its own unique characters as well. So I look at this as potentially a cell type that is multipotent, that can go either direction, depending on whatever kind of stressor it finds itself in, such as chemotherapy. So some chemotherapies drive things to basal, some chemotherapies drive things to classical. These cells are primed to go either direction. And sure enough, this is what you see, that the intermediate score for or the hybrid score, whatever you want to call it, in the Black patients is statistically higher than the white patients. So now, like I said, you have to go validate. We didn't want to keep doing this with one marker at a time. I wish that could say this using multiplex immunofluorescence, or what people are calling spatial proteomics now, is any cheaper. It's really not. But we have created a panel of 52 markers that we can use in a multiplex platform, looking for protein that we can use for validation of our spatial transcriptomics. And we're only in the beginning of this. I'm going to point out that some of these markers are for immune cells, because we definitely are interested in what's happening in the microenvironment, but also the tumor subtypes can be identified by groups of these markers, and then other fibroblasts, et cetera. And just to show you a few examples of what this looks like, this is actually a longitudinal study that Nina and I are working with, looking at primary tumors and then the lung metastasis from the same patient. These are pretty rare samples to get, so it's another reason we need to mine these particularly rare samples for every piece of data we can get out of them, which is why we use these really expensive multiplex systems. And you can see that the different characters based on TFF1, again, classical. You see the metastasis is very classical. And then the primary doesn't have a lot of anything, but got a 6. It's got some primary character, although a slightly different marker to associate it with it. And just going through these really quickly, they're again very heterogeneous, but they're certainly very informative. We think that it's going to be great when we get this all into the bioinformatics people's hands to really tell us what the data are showing, which they are actively doing now. So with that, I would give you conclusions and future directions. So in our hands, the Black African-American PDAC patients exhibit less classical behavior and an increased intermediate subtype. And what we are doing and ongoing, of course, we're going to do the validation that I told you is absolutely critical for these studies, including expanded beyond just these 15 or so patients that we've been doing the spatial transcriptomics on. We have hundreds of Black and white patients in our biobank. And so as we can afford it, we're going to continue to validate some of these studies or invalidate them, as the case may be. We have done whole exome sequencing, which we're going to use for more accurate ancestry estimation, not relying on self-reported ancestry. That is already been done, but is being, for lack of a better term, deconvoluted again by our bioinformaticians. But most importantly, I think what we're going to do is we're creating the Black African-American single cell atlas, where it's no longer going to just be 3 of these patients represented in this giant cohort. So we have gotten funding to do this, as I'll mention again in a second. We do this not because we think it's going to be, we don't know if it's going to be different. But remember, we use this atlas to deconvolute the Black and the white patients in the spatial transcriptomics, and we don't know if that's fair. We used essentially white patients to deconvolute the Black patients, and that might not be valid. So we're very dedicated to creating these databases so that the entire field in pancreatic cancer can benefit from it, and to fill the gap of what we feel is really critical for moving the biology and the science forward in this really awful disease. Of course, we're also looking at the stromal interactions between Black and white patients. We do see immune differences in the Black versus white patients, and particularly the Black patients appear to have more B cells than the white patients, and we don't know what the exact meaning of that might be, but here we go, find out. And then we also, like I said, want to do these longitudinal studies looking at the tumor microenvironment and the primary tumor, or sorry, the tumor cells themselves in the primary tumor and their metastases over time. So we actually have some longitudinal patients where we have their diagnostic biopsy, their surgical resection, and their metastatic biopsy all taken at various times during the progression of their disease, which we think is going to be very informative. So this is where I want to have the special thank you, particularly in this environment. So the Lustgarten Foundation has provided the funding for all of this, including what's coming, the single cell African-American atlas, that they've been very kind with this. And Lustgarten has been very dedicated to this particular question of the racial disparities in pancreatic cancer, in a time where we're not sure we're ever going to get funding again for such a topic. This has been incredibly important to us. So with that, I'm going to bring on the next speaker.